No matter how difficult yesterday was, just know that today is your day. Stay positive at every moment of your life. A very warm good morning and a hearty welcome to all for the second expert lecture of the academic year titled Forensic Epigenetics in Criminal Investigation. Prayer is the key that unlocks all the storehouses of God's infinite grace and power. With these thoughts, we will begin this expert lecture by invoking God's blessing. Request the Christu College Choir to lead us in a prayer song.
You made the mountains and the sea, measured out the universe, and you made me. Echoes of the voice that called the words to be, reach throughout the ages and now speak to me. You might create a king. You who made the valleys and the skies, displayed your love on far horizons and before my eyes. You who lit the stars and set the dawn in time, called them all by name, and now you whisper mine. You might create a king. And who am I that you are mindful of me? And who am I that you sent your love on me? And who am I that you are mindful of me? And who am I that you sent your love on me? You might create a king.
Epigenetic modification refers to the changes in gene expression that are not caused by changes in genetic material. The application of epigenetic techniques to legal issues and criminal investigation is known as forensic epigenetics. Current efforts to develop forensic epigenetic techniques are focused on four issues in criminal investigation: body fluid identification, individual age prediction, distinguishing monozygotic twins, and potentially stain age prediction. Because there is no cross reactivity between body fluids. This method provides greater specificity than current techniques. It is also compatible with current DNA analysis methodologies, allowing for a quick and simple interpretation. Hence, forensic genetics is a new tool for forensic detectives, and understanding it is becoming increasingly important. In light of this, the purpose of today's guest lecture is to provide a scientific understanding of current practices of forensic epigenetics, as well as the strengths and limitation of the interpretation. We are delighted to welcome Dr. Sonal Jain for the second expert lecture of the academic year. Ma'am, we deem it an honor to have you in our midst. We thank you for accepting our invitation. We welcome you, ma'am. I also take this opportunity to acknowledge the management fathers of Christu Jayanti College, Bangalore, ably led by the Principal, Reverend Dr. Augustine George, and Vice Principal Father Lijo P. Thomas, who have always led by example and ensured to provide a proactive and vibrant environment for the students to learn. I also welcome the Dean, Faculty of Sciences, Dr. Callistus Jude, and Mr. Don Cairo, the coordinator of the Department of Forensic Science, to the session. Welcome, sirs. I also welcome the faculty members of the department and thank them for their hard work and dedication towards everyday progress in the department. Welcome, ma'am and sir. I also specially welcome the participants, our dear students of the Department of Forensic Science to the session. We hope this session will enlighten you and provide you deeper insights in the field of forensic epigenetics. Welcome all of you. Without further ado, I request Dr. Suchita Rawat, Assistant Professor, to introduce the chief guest for today's lecture.
Thank you, ma'am. It is a privilege for me to introduce to the audience an active, young, and resourceful personality. Dr. Sonal Jain is a well-known academician and researcher. Ma'am earned her PhD in biotechnology from India's most reputed institute, IIT Roorkee. During her PhD studies, Ma'am was awarded a coveted Fulbright visiting scholarship to Johns Hopkins University in Maryland, United States. Ma'am was a SERP and PDF postdoctoral researcher at the AIMS in New Delhi. Ma'am has won honors like the Young Scientist Award at GenPro 2015 and the Patel Grant in 2015, to mention a couple. Ma'am has a plethora of experience instructing at many famous academic institutions, including MIT University Noida, CTM IRTE in Faridabad, Haryana, and Galgotia's University in Greater Noida, Uttar Pradesh. Ma'am also served as a visiting lecturer at the NFSU Delhi campus. At present, Ma'am is a DST Inspire faculty member at the University of Delhi in New Delhi, where Ma'am works on DNA methylation and DNA phenotyping. Ma'am's scientific interests span forensic genetics, evolutionary and population genetics, paleogenomics, forensic zoology, and medical genetics. In numerous national and international conferences, Ma'am has presented her research finding and has published many peer-reviewed research articles in Index Journal. With this reputed career, we feel it an honor to have Madam in our midst to address the second expert lecture of this academic year. I now request Ma'am to address the gathering.
Thank you so much, Dr. Suchita, for the invitation, and thank you, Ashika, for welcoming me. I'm also happy to be here. So, shall I share my screen for the presentation?
Yes, ma'am, you can do that.
Okay, just. Just is it visible?
Yes, ma'am, it's visible.
Is it fine?
Yes, ma'am, it's perfect.
And am I audible to everyone perfectly?
Yes, ma'am, you're audible.
Okay, thank you so much. So I'm so happy to be here, and I usually would like to enlighten first the UG and PG students, and who are my audience actually. So just to start with my lecture, I would like to address few points. Like the topic forensic epigenetics in criminal investigations. This is one of the hot topic, not in India but all over the world for the research purpose. There are certain problems which currently India and other world criminal investigation agencies are facing, and that can be resolved through forensic epigenetics. So basically, as Dr. Suchita introduced me, I have worked during my PhD on ancient DNA, and I have an experience in like how to extract the oldest of DNA. And we have extracted DNA from 25,000 years old samples. And to continue with that, I did my PGF from AIMS, And then I taught in various institutes. And then I realized that research is my first love. So I just applied for this fellowship, DST Inspire Faculty, which is a 5-year project. And currently, as she told, I am working on DNA phenotyping and DNA methylation. So DNA phenotyping is a part of forensic genetics only, but DNA methylation is a part of forensic epigenetics. I don't know why it's not moving ahead. Okay, it's with the space bar. Okay, so just excuse me for the my sore throat because of the weather in Delhi. So I might be coughing in between. So before starting, I would just like to say to the students of BSc and MSc, to the BSc students, they should also look for, if they are interested in the field of DNA, they want to go for forensic DNA analysis, or the MSc students who are interested in the field of genetics, then this lecture may give you certain points which could be helpful for your research if they want to pursue their career in research. So this will shed light on few points which can be taken up as a research career. So as we all know, genetics is basically the study of genes and heredity, and as these traits which are passed from parents to offsprings, which is a result of changes in DNA sequence. But as also Ashika told, that epigenetics, it is the study of how your behavior and environment can cause changes which will affect the way our genes work. So basically, epigenetics, if you can see this image, the genetics part, it is the DNA sequence variation which will lead to gene regulation and the phenotype will appear. For example, an offspring's DNA is 50% DNA of mother and 50% DNA of father. Depending on the traits which the offspring has acquired, our phenotype will appear like this. Depending on which eye color we acquire, which skin color we acquire, which hair color we acquire, and other things which are hereditary transferred from parents to the offsprings. But epigenetics, what is the role of epigenetics here? The environment, there are many factors. For example, the diet, other environmental factors like pollution, or the other drug abuse, or smoking, or alcohol, etc. All these factors, these are known as, if they modify, the modifications produced by these factors are known as epigenetic modifications. And these modifications, they also regulate the gene, which will also again appear in our phenotype, and that can also be measured. DNA methylation, histone modification, and non-coding RNAs. These are the 3 types of epigenetic modifications. Before going to what is forensic epigenetics and forensic genetics, I would like to make you aware what are the different types of epigenetic modifications. We can say in simple words, epigenetics is on the top of genetics. It is the level of control that sits above the genes and determine which genes are turned on and which genes are turned off. And these turning off and turning off is reversible. Just remember this thing, it is reversible. So the 3 factors which we have discussed in the previous slide, DNA methylation, we have this DNA methylation, we have histone modifications, and we have these non-coding RNAs, which are responsible for these epigenetic modifications. DNA methylation, in simple terms, if I say, the adding of a methyl group. As you can see in this figure, there is this base cytosine. As we know that our DNA is made up of 4 different bases. So this cytosine, if at this position, the addition of methyl group happens, then this cytosine is known as methylated cytosine. So we can say that DNA methylation has happened at a particular space. For example, in our DNA, if at a cytosine at a particular position, a methyl group is being attached, then we can say the DNA is methylated. So as you can see in this there, the addition of a chemical group to DNA is DNA methylation. And this group is added to a specific places on the DNA where it blocks the proteins that attach to DNA to read the gene. So now we can say when this methyl group is added to that particular point, it blocks the proteins which will be attached to DNA, and therefore the proteins cannot read the gene. So therefore, how the function stops because of the adding of methyl group on the cytosine, the function of that gene stops because the protein cannot read that gene. This chemical group can be removed through a process called demethylation. So in our DNA, at various points, methylation and demethylation can occur depending on the diet we are having, the environment which we are living in, or if we have any other drug abuse, etc. So this technique, this process of methylation and methylation may turn genes off, and the process of demethylation, if it, as I told you, this process is reversible. So if demethylation happens, it turns the genes again on. Histone modifications. We all know that DNA wraps around proteins, they are called histones. Similarly, again, in histones, if some genes are wrapped around histones and are turned off, while some genes are not wrapped around histones and are turned on, the chemical groups can be added or removed from histones and change whether a gene is unwrapped or wrapped. As you can see in the next slide, see, this, if we say this is a histone protein, and the red mark shows that methylation is happening, the green mark shows that acetylation is happening, the blue mark shows the phosphorylation, and this brown is ubiquitination. So the four process can happen in the histone modification. Similarly, as I told you, for the DNA methylation, if there is any modification in histone, then it may also stop the gene function. So it can turn the gene off as well as it can turn the gene on. The third is the non-coding RNAs. The protein is made by the coding RNA, and the non-coding RNA, it helps in controlling the gene expression by attaching to the coding RNA, along with certain proteins to break down the coding RNA so that it cannot be used to make proteins. Non-coding RNA may also recruit proteins to modify histones to turn genes on or off. Just excuse me. Okay, so DNA methylation, and then we have histone modification, and then we have non-coding RNA. These three epigenetic modifications, they may modify in certain ways to make our function of genes either turn on or turn off. This non-coding RNAs makes up the 80% of transcriptome, and the coding RNAs is almost less than 3%, which mRNA codes for the proteins. And in this non-coding RNAs, we have small non-coding RNAs, which are less than 200 nucleotides, and more than 200 nucleotides, we call them as long non-coding RNAs. This is the list. This is for basically, if you are totally into the core research, then we have to go for it. Then we'll come to the main topics. So the thing is, if we are talking about the forensic epigenetics, and we have to capture what changes are there in the DNA methylation. So we have to know how this epigenetics is making changes in our body. For example, if we say the first point, from the beginning of the birth, or the epigenetic changes begin before even we are born. All our cells have the same genes, but they look and act differently because of epigenetics. As we grow and develop, epigenetics determine which function a cell will have. For example, whether it will become a heart cell, nerve cell, skin cell, etc. So the role of epigenetics starts before even a child is born. The second is epigenetics and age. A very simple example I want to give you. Supposedly there are two persons who are of the same age, 30 years old, and they say, you don't look your age, and someone say, you look more than your age. So this is what the epigenetic is playing. Their chronological age is same, but their biological age is different. That may be due to different reasons. You can say the person, he is looking more aged because of various reasons. Supposedly, he is having an early graying of hairs. which is due to the environmental factors. Nowadays, many youngsters within the age group of 18 to 20, 18 to 30 years, young adults, we can call them, they have early graying, which is due to the pollution or the other environmental factors, and which is the phenotype which makes us look more aged. So the epigenetics change throughout our life. Our epigenetics at birth is not the same, which is during childhood or adulthood. For example, there was a study conducted where a newborn DNA methylation and a 26-year-old and a 103-year-old person's DNA methylation, level of DNA methylation was calculated. We have seen the highest level of DNA methylation was present in the newborn, and the lowest was in 103 aged person, and the 26-year-old person had a level of DNA methylation between these two individuals. Epigenetics and reversibility. Not all epigenetic changes are permanent. Some epigenetic changes can be added or removed in response to changes in behavior and environment. A very good example I would like to give you. Nowadays, everyone is suffering from various lifestyle diseases. A person who is a younger adult or an older adult, more than 30, hypertension, you can say. So many people are dying because of early heart attacks, or they are having hypertension at a very early age. So many females, they have irregular periods, or they have PCOS, PCOD, or infertility. All this issue, if we talk to doctor, they say these are the lifestyle diseases because of what we are eating, the obesity, which is increasing nowadays in our country. So all these are epigenetic modifications, and if you regulate your diet, if you try to little bit change up your environment, and if you work on your body, if you do exercise, so all these changes can be reversible. So all the diseases which are related, the lifestyle diseases which we call them, is basically the epigenetic modifications which are happening in our body. They are turning some gene function to off, and some diseases resulting into some diseases, which we call them as lifestyle diseases. Smoking can result in epigenetic changes. For example, the gene AHRR, the smokers tend to have less DNA methylation than non-smokers. This part in our lab, we are working on this to see what are the epigenetic modifications happening in a smoker and a non-smoker. The difference is greater for heavy smokers and long-term smokers. After quitting smoking, former smokers can begin to have increased DNA methylation at this gene. Eventually, they can reach levels similar to those of non-smokers. That means reversibility is there. Now, the epigenetics and health. Germs can change our epigenetics to weaken our immune system. For example, Mycobacterium tuberculosis, which causes TB. Infections with these germs can cause changes to histones in some of our immune cells, which results in turning off the IL-12B gene. And these result in the weakness of our immune system and improves the survival of Mycobacterium tuberculosis. Because as doctors generally say that your immunity is weak, if you improve your diet, then your immunity may increase, and you will have a better immune system. So somehow these things are related. The other is the cancer. Epigenetics can also be used to help determine which type of cancer a person has, and can also help to find hard to detect cancer earlier. We will be talking about one part of genetics, which is forensic epigenetics. But if you see, there is a lot of research going on about the hypertension, diabetes, cancer, and other diseases related to epigenetics in India as well as in world. Various institutes like AIMS, ICMR, and other PGI, they all are working on this epigenetic analysis of various diseases. This is also nutrition during pregnancy. This says that, as I told you, that the environment and behavior during pregnancy also determines that what certain diseases the child may get. So there was a study in the Dutch hunger winter famine. There was Dutch hunger winter famine, and a study was conducted where mothers were pregnant during this time. They were more likely to develop certain diseases such as heart diseases, schizophrenia, and type 2 diabetes. And these people had increased methylation at some genes and decreased methylation at other genes compared with their siblings who were not exposed to famine before their birth. So you can say the epigenetics play a role, since even before we are being born. Okay, so the previous discussion or the previous part I have told, it's about how the epigenetics has a role in determining our health. Now, what is the applications of this forensic epigenetics in criminal investigations or in our legal system? We all know that the one important major task as forensic scientists, it is to solve a crime, and that is how being done by identifying an individual. Our major task after all the analysis is the identification of a criminal. We have many evidences which can be found at the crime scene. For example, starting from the uh, fingerprints or the document evidences or any evidences which are any chemical evidences. But the major evidence through which we can the directly link the crime scene or the crime to the person is the biological evidence, because the biological evidence contain certain cells or tissue which can be analyzed and a person can be identified or individualized. Okay, but we have to remember one thing here, that most of the forensic genetics determination nowadays is totally dependent on STR profiling. The criminal profiling is totally comparative. That means we need to have a person's DNA, or we need to have certain suspects whose DNA can be compared to the crime scene's DNA. So as I wrote the project which I am working on, this is one of the major problem which I came across, that as DNA, as we don't have a DNA database in our country, although we are working on it to develop a whole DNA database with CCMB and CDFD, they are working on it. So without having a DNA database, we don't have any sample for comparison if we don't have any suspects in that case. So what is the solution for that is the DNA, forensic DNA phenotyping, where we can work on if we have any person's DNA on the crime scene and we don't have any DNA to compare, then we can look for that how a person, what would be the person phenotype, looking at the details of those DNA. Okay, so currently STR profiling is the major various forensic DNA labs. They are using tandem repeats the STR DNA profiling for the forensic genetics analysis. And the SNP's research is slightly developing with various researchers from CDFD and various researchers from University of Pune, and now University of Delhi is working on the forensic DNA phenotyping. So this forensic DNA phenotyping is developing in India with a great pace. We know that forensic genetics, here we use genetic analysis to answer questions of interest to a court of law, and is largely centered around the characterization of hypervariable polymorphic regions referred to as STRs. But whereas if we see the forensic epigenetics, it is the use of epigenetic techniques, which we have already discussed, like the DNA methylation, microRNA markers, which are basically non-coding RNA markers, histone modifications, to address the question of interest to the court of law and the criminal investigator. So with DNA in the forensic genetics, if we have DNA of a person, what all you can identify and which all questions we can solve. We can talk about if you see the first thing, we can talk about the human species identification, first thing. We can talk about the ancestry information. And as I told you about the forensic DNA phenotyping through the SNPs, like SNPs, which is single nucleotide polymorphisms, various traits, various single nucleotide polymorphism is responsible for various traits. And we have to look for it and we have to analyze the DNA for the same to develop a profile of a person for his or her identification. And then with the DNA, we can go for age prediction. So my project is, we are currently working on DNA phenotyping as well as we are working on the age prediction through DNA methylation. Okay, so what is the need of this study, whether the DNA phenotyping or the forensic epigenetic study? As we do not have, if we go about the NCRB database of 2018, there are so many pending cases. We don't have any DNA database. We don't have any investigative lead in the case of unknown samples because we have nothing to compare with. I would say that no single case should remain without justice. Everyone should get justice. So even if the technique of SNP phenotyping or the technique of DNA methylation will help in solving even few cases, then I will feel like that we have success in this field. Okay, so the literature gap I was talking about is because of the absence of DNA database, absence of samples for comparisons in the cases of unknown samples, and slow tracking of cases. Because of overpopulation, our forensic services are overburdened. So we need a fast method to solve the cases which are pending. We should know what is the reason of the pendence of cases, why they are pending, and we should solve the reasons for that. So phenotyping and DNA methylation may be the answers for those cases which are cold cases, and in those cold cases we have no investigative leads. Interestingly, in the last 1 month, DNA phenotyping, which is, I'm just telling you, it's taking up a pace in the whole world. So one of the lab in USA, they have developed this technique. We are still in the process of developing this technique, but in USA they have developed, and they have solved 2 cases. One of the year 1972 and one of the year 1984. Such old cases they are able to solve because of the development of this technique of DNA phenotyping and DNA methylation. These are the, Ashika also told that the major applications of forensic epigenetics. So these are the 4 major applications of forensic epigenetics. The first is the body fluid identification. Second is individual age prediction. Third is differentiation between monozygotic twins. And fourth is day age prediction. So the forensic investigation, we have basically few questions to answer. Who did it? Human identification, we have to identify the person. Who was the person who did this? Who has committed this crime? That means to identify the individual. Through investigative leads, we can know about the age of the person, as shown by this, the ancestry of the person, and how the person appears. This, I won't say that the answers given by the forensic epigenetics or the forensic genetics or the forensic phenotyping can make you catch hold of the person 100%. But we will have certain investigative leads that it may lead to a person, and conclusively it may lead to a person depending on the what other evidences are available with us. So after determining who has committed this crime, other questions we have: what was the biological evidence, or what was the stain which was there at the crime scene? What was that stain? Which biological fluid was that? Which tissue was that? So through DNA methylation or through forensic epigenetics, we can determine or we can identify the stain. And how? The third thing is how the crime was being committed. So in the body fluid identification, we have blood, various body fluids. The major, if we talk about blood, menstrual blood, semen, vaginal fluid, saliva, etc. Now, the question is, what is the difficulty? Nowadays also we are identifying body fluid identification, we are doing through various methods. But the methods which are now being used in the various forensic science lab, for example, various chemical tests we are doing, we say these are the preliminary tests, or we say these are the confirmatory tests. But all these tests, they have certain limitations. Some tests may give false positive. And in the cases when the body fluids are too old to give any test, or they are contaminated with any other surface, for example, any other contamination, any other contaminant is there. So it is very difficult to identify that particular body fluid, or it is difficult, or the test may give false positive. So for these, we have to go for the forensic epigenetic analysis, which can be conducted through microRNA analysis or DNA methylation analysis. For the blood, I would say that these are the microRNA markers which are being identified by various studies. For venous blood, we have this various microRNA markers. For saliva, we have these microRNA markers. For semen, we have these markers, vaginal secretion, menstrual blood. These are specific to these particular body fluids. Okay, so how to go about it? Here I would like to add one more point. The best part about this is, if we have any biological fluid on the crime scene, from this technique, we can side by side extract DNA, as well as we can conclusively determine which body fluid is that. So as you can see the flow chart, we have the unknown. If we have the unknown biological sample, we can go for the extraction of DNA RNA content. From the DNA content, we can go for STR-DNA profiling, which will help in the person identification. And from the RNA content, we will use the microRNA markers, which will help in the determination of which body fluid is that. So for the extraction techniques for microRNA markers, we can use stem loop technique or poly A tail extension. This is, if we go in detail, we can have another discussion on these for the detailed analysis of these microRNA markers. Microarrays can be used, quantitative RT-PCR can be used, RNA sequencing, and capillary electrophoresis. So of the basically it's just like we have to extract the RNA and DNA. DNA can be used for the STR analysis, and from the RNA we can look for the particular microRNAs. For example, if microRNA which you have observed is microRNA number 372 and 617, if you see the peaks of these two microRNAs, then you can conclusively say that this is a vaginal secretion. A very interesting thing I would like to add upon here. Obviously, these techniques are much more costlier than our day-to-day techniques of chemical identification of these body fluids. But if a person is suffering from any diseases, supposedly a female, if we have vaginal secretion, if that female is suffering from a disease and some fluid is being extracted with her vaginal secretion, then that particular disease can also be identified. For example, if a person is also, if a male is suffering from any disease and that particular chemicals are found in his semen, then just imagine how easy it is to identify that particular person. If you have certain suspects, and if you don't have any suspects, then it will give you such a good point for individualization that this person is suffering from that particular disease. So depending on which microRNA is present, we can identify the body fluid. For tissue differentially methylated regions, we have this CG sites. These are basically DNA methylation sites. The previously which we spoke about the markers, those were non-coding microRNA markers. Now we are talking about the DNA methylated regions. So different tissues have different DNA methylated regions. For example, if I tell you, supposedly if I have blood, and if I have to identify the age of a person using blood, then the markers will be different. And if I have to identify the age of a person using saliva, then the markers will be different. That means different tissues have different markers for identifications. These markers, as you can see here in this table, we have a shortlisted few markers. If you say CG is basically the site, that site where methylation has happened. Okay, as I have told you, if you just remember, just try to remember it. If we just imagine the DNA at the cytosine, methyl group is being added, that is DNA methylation. So at that particular site where where the area the methylation happens, we call it as the island, CPG island. That is cytosine, which is followed by guanine. That is the site, basically CG, cytosine, and guanine. That is only cytosine which is being followed by guanine, their methylation will happen. So that is how these markers are being named as like this. So blood have these markers for identification. We have these markers for identification for semen, for saliva, for menstrual blood, for vaginal secretions. Okay, so as we have discussed about the microRNA marker technique in this slide, that we have unknown biological sample, then we will extract DNA, RNA. Similarly, we have unknown biological sample, we will extract DNA. We will, after extraction of DNA, we will go for the bisulfite conversion of DNA. After bisulfite conversion of DNA, we will go for PCR, and then we'll go for the pyrosequencing to know how much methylation is there at the site. Now the next thing is, which is also now a very major problem in our forensic investigation, if we have the mixture of body fluids, then what can be done? For the mixture of body fluids, as you can see, if the body fluids are mixed together, then there are, as the markers of all the body fluids are different, then they can be analyzed using Malditov technique. Or Malditov is being combined with microspectrophotometer, where we have peaks of different body fluids, and then we can identify that in this particular mixture, which body fluid is present. Depending on if we have peak of semen and vagina, then we can say that this mixture contains semen as well as vaginal secretion. Supposedly that mixture also contains saliva, then we can conclusively say. So the one most important point is, if we have found that particular marker and you have identified that marker, then this is 100% conclusive result to say about the body fluids and the mixture of body fluid identifications. You also have the conclusive result of the same. Now, second major application is the individual age estimation. Individual age estimation is usually done by DNA methylation. Recent epigenetic investigations have found that there is a very close association between aging and DNA methylation. Various studies have been done in this prospect. The first study was conducted, I think, is in 2011, and we have this epigenetic clock through which we can identify the age or estimate. Basically, we'll say estimate the age of a person. DNA methylation is the best understood epigenetic signature in the genome. With increasing age, DNA methylation patterns gradually show a genome-wide hypo and hypermethylation. That means the studies have shown that at a particular age, the DNA methylation levels will reach a peak. Till the age of 55, the methylation level will increase. But after that, we have observed that the methylation level gets a drop after the age of group of between 55 to 60. These variations emerge before adulthood and thereafter they increase. And the epigenetic patterns increase exponentially up to adulthood, and then they slow down in a linear pattern of change later in life. And the best part is that they can be calculated, which helps us in the estimation of age. So this is the same thing. We have cell nucleus, chromosome, and the DNA, you can say, is C in between, and the addition of methyl groups on the C, which is followed by guanine, CG. That's why we call them as CPG sites. So you can say in this, the younger DNA, you can see the more methyl groups are added, and in the older, you have less methyl groups. This is the same as we have already discussed. The addition of methyl group to C nucleotide, that particular site is known as CpG site. It is an epigenetic marker which is involved in gene regulation, occurs throughout the genome, and the patterns at certain CpG sites are linear over the time, which can be calculated and which helps in the estimation of the age. And these different samples, different tissue samples have different methylation levels. So if you are going for a research, then you have to see. If you are going through blood, then it will have different methylation levels in the CpG. The CpG sites have different methylation levels, and the saliva will have different, semen will have different methylation level. This is the various markers or the genes which are responsible or which have CpG islands which are involved in the age estimation. For blood, we have genes, for example, SPA, PDE, 4C. So with the literature or with the research we are doing, we have got to know that ELOVL2, FHL2, and one gene CLORF2 that is responsible for, the 95% of accuracy is observed in their DNA methylation patterns. So basically, we follow the same technique: extraction of DNA, bisulfite conversion for age estimation. Then we select the CpG sites and biomarkers. Library preparation and quantification through biosequencing, data analysis, age and statistical analysis, make a computational model for the estimation of age of an individual. Then we have the third aspect, which is the monozygotic twins. With the forensic genetics, as we know, the monozygotic twins have the same DNA, exactly same. So there are many cases. There was a famous case where the twin was caught because the crime was conducted by the different twin, but as the DNA matches with the other twin, the different twin was caught, and he was punished for the crime. So in such case, the forensic epigenetics, it comes in forefront. We have been studying the DNA methylation levels varies with the change in environment, with the diet we are having. So the same methylation and gradually show more differences in the methylation patterns. There are specific forensic markers which can be used for the identification for the same differences in the line 1 in interspeed repeat sequences. These are the microRNA markers. There can be some environmental influences, for example, smoking. And the monozygotic twins, they usually have a similar growth environment. We can see one CpG site with DNA methylation was significantly different in all twin pairs, and the highest number of significantly different CpG sites were 6. That means 6 CpG sites have been identified by Richards et al. In 2019, which conclusively said that with the change in the environment or the lifestyle, the twins have DNA methylation patterns which can be calculated and which helps in the identification or the individualization between the two twins. As you can see, the twin A and twin B, they have it is being represented. If you see these arrows, they are being represented as the methylation. So you can say at the telomere, they have almost same DNA methylation patterns. But with the increase in age, you can see the methylation patterns changes. Supposedly the third stick here is white, or here the second and third stick is white. This epigenetic clock, this has certain influence from the early life till the old age. And as the age grows, the epigenetic markers which are present in our body, it changes from person to person. Thus, through just these epigenetic markers, maybe with the identification of LTR, sign, line, these are the generalized hypermethylation markers, or with the CpG sites, we can really identify and individualize two different twins. And NGS or the MPS are the techniques which can be used for the identification. And one interesting study in the 2015 by Stewart et al., they have demonstrated the use of high resolution melting curves analysis, which can easily differentiate between the monozygotic twins. The older the twins, the more different are their melting curves because of the difference in their epigenetics or the epigenetic modifications. Now, the one of the more fourth important application of forensic epigenetics is stain age identification. That means how old was the body fluid which was left at the crime scene? What is the age of the stain? This is a very new research which is being going on, and conclusive results have been observed in various researches, but still we have to develop and form this technique for the 100% conclusive results. There are certain limitations which come with the stain age identification. Currently, one project we took up for our MSc students, we identified the age of stain using the UV spectrophotometer. It also gave very nice results. But for the conclusive results, we can go for the stain age identification. We can go for the various genetic markers which are present there. As the body fluid leaves the body, there will be changes in the oxygen environment, and the degradation rates of these genetic markers are being studied for the identification of these stains. For blood stain, Anderson et al. And in 2005 and 11, they had done these studies. Semen-specific markers were observed by Board et al. In 2019, and RECFA and HIF1A, Asagir et al. 2019, they have certain specific markers for the stain age identification. The major limitations which come, or the major challenges which come for the stain age identification, is the extent and influence of the factors altering the markers. Supposedly, if we are not aware of what are the environmental factors our body fluid stain has come across, then we cannot say conclusively like whether the markers have degraded with the time which has been spent, or it is the environmental exposure which is showing the degradation of these markers. So it gets really difficult to use this data to form a useful opinion. These were the four major applications, but there are certain current and future prospects which forensic epigenetics or forensic epigenomics can solve in the future. We can say about which tissue, how old a person is, which twin, whether the person smokes or not, alcohol intake, whether the person takes alcohol, how much alcohol does he take, any drug abuse is he going through, body size and shape. It can give clues on diet, how active that person is, because as I told you, the reversibility, the obesity and the body shape, etc. can be changed if you become more active or you don't follow a sedentary lifestyle. Then your DNA methylation markers can be reversed to where the person is living and the socioeconomic status of the person, which can or cannot be tell. So depending on how much information is available to us, then the forensic epigenetics or the forensic epigenomics can play a role. This particular figure is from Vidaki et al. 2017. I forgot to mention the reference. So the applications which we have discussed is which tissue, blood, menstrual tissue, menstrual blood, semen, vaginal, saliva, which body fluid is there, how old the person is depending on the methylation levels, whether the person is a smoker, whether the person takes alcohol, whether the person is illicit drug abuse. So by DNA methylation pattern, we can shed light on all these factors. Although coming about all the applications which we have discussed about in the forensic epigenetics of the DNA methylation and other non-coding markers, there are certain challenges which comes with it. For example, various ethical and legal issues. Supposedly, if we talk about the gender of a person, or the person if he is smoking alcohol, or its age, or the phenotype prediction, then they come as the privacy issue, or supposedly any link to disease risk, or another major challenge could be the low quantity or quality of DNA. We really need nanogram picogram level quantity of DNA for the analysis of this. The consideration is that for the same that high sensitivity, limited number of CPGs, multiplexing assay design, and for the validation of that. For methylation, we need to convert the DNA into bisulfite, and amplification bias could be there, but it has the robust applications. For marker discovery, we need large data set. We need increased number of CPG sites, and which need to be replicated. We need appropriate data analysis, and we need tissue specific patterns. And for tissue specific patterns, we need their matching samples or their particular microRNA markers for their identification. So there are with every technique which is being developed, and for various future aspect research, there are certain challenges and considerations which need to be overcome with time. So in the concluding remark, I would say that this field is gaining a considerable amount of importance from past few years within the community. This is basically a potential to be, has the potential to be paradigm shifting in terms of progressing criminal investigation. It will help us in enhancing the amount of information obtained from a crime scene, but will also help in addressing the challenges to the evidence that can be made in the court of law. Thank you so much. Okay, back to Dr. Suchita.
Thank you, ma'am, for the engaging and profound session. If the students have any question, they can unmute and ask the questions one by one.
Don't hesitate to ask anything. I have kept the lecture as simple as I can make it for your better understanding. So if you have any doubt related to anything, you can go ahead.
Thank you, Dr. Sonal, ma'am, for this wonderful presentation. I'm sure they have a little bit question, but the presentation itself was so self-explanatory that a lot of the doubts were cleared. Ma'am, two questions from our end. One is, where do you see the legal admissibility of forensic epigenetics when it plays to Western versus Indian scenario? What about the legal admissibility of this technique?
Okay, so currently, I would say the passing of DNA bill in Indian scenario was also, it took a lot of time, and still we have certain issues with the DNA bill pass. So coming to, still we are struggling with the developing of forensic epigenetics in India. As per my knowledge, I mean, very few people are working on forensic epigenetics in this field. Coming to the admissibility of that evidence in the court of law will be the next question. First is the developing of a robust system of the analysis of forensic epigenetics is the first thing. So I think as the forensic epigenetics methods will develop better, then we will talk about the admissibility. It will, I think, at a certain point of time, we will have some issues, because this will talk about the disease of a person. This will talk about the phenotype of a person. This will talk about the various gender issues. So I think at certain point of time, there will be queries and there will be legal issues, but we'll see how it will go about that.
Thank you so much, ma'am. Ma'am, there's another question. Is there any particular case where forensic epigenetics has been very useful? And we're talking about in general, not just India, but then other foreign countries as well.
Yes, so many. I mean, all these researches which I have just mentioned, all the microRNA markers I have mentioned, they are being they have these sets of snapshots or the genetic marker set which are being used in all over the world. They are being used. So there are so many cases which are being solved. So as I was talking about the forensic DNA phenotyping in USA, so their their evidence is admissible in court of law, and they have solved this case on this basis. But as I'm telling you, in India we are still establishing these techniques, so it will take a little bit of time to be admissible in the court. But side by side, I think the the technology which we are using is totally scientific. So I think there should not be any issue. The cases, there are so many cases all over the world, in Europe, in Asia, in America, where they have used this forensic epigenetics for identification. They are this age estimation. They have this by Hanuman et al. And Harvard et al. They have this epigenetic clock which they have made, and whatever sample which comes for analysis, they go for the age estimation, so that they can shortlist the number of suspects. Supposedly, if you have like 15 suspects having various ages, so they run their sample through the epigenetic clock estimator, and which will shortlist their sample size to supposedly the persons who are aged between 25 to 30 years. So that will help, obviously, in proceeding in a better way.
Thank you, ma'am, for that answer. Ma'am, we have one
Ma'am, I
question.
have a question.
Yes, Oliver.
Ma'am, you mentioned about the mixing of fluid, right? I didn't
Yes,
really
yes.
get that method, how the markers thing, how do they differentiate it?
Okay. Which Oliver, you are BSc or MSc student?
Ma'am, I am BSc student, first sem.
Okay, first sem. So have you heard about the techniques like FTIR, fingerprinting techniques, etc.?
Yes, ma'am, I've heard about fingerprint.
Yes. So this not fingerprinting, like FTIR technique. So like the other techniques, supposedly if we're talking about the FTIR, if we put any sample, analyze any sample in the machine of FTIR, the sample, supposedly if you say the sample is of it's mixture of various compounds. Supposedly it has methanol, ethanol. So what you will see the ethanol is C2H5OH. So you will see carbon, hydrogen, OH group, right? So ethanol is C2H5OH. Carbon you will see, hydrogen you will see, OH group you will see. Similarly, in this case, if you have the mixing of samples, supposedly there is a rape case, and the swab has been taken from the victim, and the swab contains vaginal fluid as well as semen analysis. So you have to check whether the female, the victim, the semen is present in that particular fluid or not. So you can analyze this, you can identify the microRNA markers when you run through the MALDI-TOF as well as microspectrophotometers. Then you have the different peaks. The peaks will show that this mixture contains vaginal fluid as well as seminal fluid. So that's how it can identify and differentiate. Okay.
Thank you, ma'am. Thank you, ma'am.
Okay.
Dr. Sonal, there is one more quick question that we have from the audience. It is
Sure.
how far SNP use is being possible in India, and does the SNPs change when we're talking about Indian consent? Because the research article that you presented were based on populations that were abroad. So is there any such finding in Indian context where the SNPs have been utilized, and are they changed compared to the Western population and Indian population?
Okay. That's a very interesting question, whosoever has asked, because the research is totally based on that, the work we are doing, because we know that our ancestry is different. If we say a European and if you say an Indian, phenotypically, if you observe a person, what changes you will see? You will say that they have a lighter hair color, we have a darker hair color. You will say they have lighter eye shades, we have darker eye shades. These are the phenotypic differences. Or you say they are more fairer than us, and we are comparatively, we have the pigmentation more, melanin pigmentation more than them. So obviously, if the phenotypic differences are more, then the DNA is also different. There are SNPs we are observing that certain SNPs are responsible for the similar SNPs are responsible for similar traits in both of them. But speaking about the population differences, the SNPs are obviously and they are different. So we can say in the way, supposedly if you talk about a particular pathway, let's talk about skin color. Skin color, if we say how the skin color is being imparted to us, that is the melanosis pathway, how much dark pigmentation and light pigmentation is there. On this pathway, there are many genes, and in these genes, there are many SNPs. So the pathways are different, SNPs are different, but certain genes, they are they are more associated, or we can say certain SNPs are more associated and certain SNPs are less associated. For example, let's take an example. There are 30 SNPs responsible for skin pigmentation. Just an example, there are thousands responsible. So 30 SNPs responsible for skin pigmentation. So maybe like 15 are more associated in European population, and the other 15 are more responsible in Indian population. So this project target is, we are working on this. We have identified the SNPs and our samples are being analyzed on that, and hopefully will come with results in the next year with which SNPs are more associated with which phenotypic traits. And the aim of this project is to make a snapshot kit of all these phenotypic traits, so that if there is an unknown sample from the crime scene, we can directly run that sample on this snapshot, and then we can have a profile of the person, like what is the skin color of the person, what is the hair color, what is the eye color, how does the face shape look like, and what is the eyebrow thickness of that person, what is the nose shape of that person, which will give a phenotype of a person, or you can say portrait of a person, portrait kind of a person. So the major answer is yes, they are different in different populations. That's why the population studies are being going on in different continents or in different countries, which are population specific studies. Even we have taken the population of North India, and for the validation set, we will take population from Northeast India and South India, so that we will see. As you might have gone through who's a population genetic expert, they might have gone through the paper of Dr. Thangaraj of the ASI and ANI, that the South Asians have different ancestry and North Indians have different ancestry. So we will compare that, like whether we have different SNPs or the South Indians compared to South Indians. This data which I am presently working on, it covers the 6 major states of Northern India. We started from Jammu Kashmir, and then we took Himachal, then Uttar Pradesh, Haryana, Punjab, Uttarakhand. We take these 6 state samples and we are analyzing our results on those 6 states.
Thank you, Dr. Sonal, for that wonderful answer. One quick last question from one of the MSc student, and then we'll close the session for
no problem.
the question. Since these students are pursuing their masters and they would want to work on with future research, out of the 4 methodology that is used, the application, which is body fluid identification, individual age prediction, monozygotic twin differentiation, and stain age prediction, which one is the best research area that they can work on? So one of the students sent this question.
I will say, like, currently we are touching the age estimation, but while I was going through the literature, and I spoke with various agencies like Thermofil, etc., for my sample analysis. So they only told me, ma'am, please work on mixed body fluid identification. They said, ma'am, we have certain certain queries from even various central forensic science labs also, where they are struggling for the identification of various body fluid mixture. So I think if you want to explore this area, this would be a very good area where you have so many things to play with. Like you can mix semen, saliva, and vaginal fluid. You can mix different types of body fluids, and you can see how they react with each other. You can also see with age how much the deterioration is there, how much markers can be identified. So that is a very research area to go with, and which have a major impact in future also. One more thing is, it would be really good to find out if in the case of a gang rape, if the victim's vaginal secretion contains multiple semen samples, then to identify each person individually, and if we can segregate the DNA and identify the person, then conclusively, if we can give the result, then this is a great area to touch upon.
Thank you for the answer, ma'am.
Thank you, ma'am, for patiently answering our questions.
No problem, no problem. Happy to be.
Gratitude is the currency that we can mint for ourselves and spend without fear of bankruptcy. With these thoughts in mind, I invite Ms. Aishwarya Shashikumar, Assistant Professor, Department of Forensic Science, to deliver the vote of thanks.
Ma'am, you're not audible. Aishwarya, ma'am.
I would call upon Devi ma'am to deliver the vote of thanks. Thank you, ma'am. Knowledge is power. Knowledge shared is power multiplied. On this note, I would like the opportunity to thank Dr. Sonal Jain for giving up your valuable time this week to speak at a program in spite of your health issues. It was a pleasure to listen to you, ma'am. It was clear that your presentation captivated everyone in attendance. We could not have pulled off such a successful event without your help, ma'am. Ma'am, your knowledge and experience can teach us so much more. Best wishes for your good health and success in all your future endeavors. This session would not be possible without the support and guidance of the members of the management, led by the Principal Reverend Dr. Augustine George and Vice Principal Reverend Father Lijo P. Thomas. Thank you, Fathers. I extend my gratitude to Dr. Callistus Jude, Dean, Faculty of Sciences, Christu Jayanti College, for encouraging us to strive better and helping us in conducting such programs. Thank you, sir. The smooth conduct of guest lecture is because of the faculty members of the Department of Forensic Science. I thank all the dear teachers for coordinating the program to make it a success, and a special thanks to Dr. Suchita Rawat for arranging this session for the department. Thank you. I also thank all the students of BSc and MSc Forensic Science for being a great audience, for their patient listening and asking their doubts to make this session interactive. Thank you, dear students. I would like to extend my thanks to the IT department of Christu Jayanti College for providing the necessary resources to conduct this event and ensuring there was no glitch all through the session. Thank you, sirs. Thank you, one and all. As we have come to the end of the program, I request all to join together for the college anthem.
Like a golden disc, sun in the rising east, lights up a life divine, fills knowledge in our minds. Laurels and the book make us sublime. With the mighty Lord, we soar high on the bedrock of faith. Joy and peace sprawl our hearts and minds. On we march our goals as a beacon for the world, with a passion for our growth and a love for humankind, united and strong in excellence on the bedrock of faith. strive. Christo Jayanti, Christo Jayanti, life and prosperity. Christo Jayanti, Christo Jayanti, life and prosperity.
I request the participants to turn on their video for a photo screenshot of the session. A feedback link will be shared to your WhatsApp groups. The student participants can fill the feedback form. Thank you so much, ma'am, for your session.
Thank you. I wish everyone all the best for their future endeavors.
Thank you, ma'am.
Thank
Thank you
you.
so much. This is Don here. There's some problem with
Yes.
my system, so I'm using my phone camera. So probably you
Okay,
won't be
okay.
able to see. So that's right. Yeah, thank you
No
very much.
problem.
It was very nice. Thank you for your time. I think even though you're not well, you have spent that time with us. So
Yeah, that's fine. I didn't want to cancel it just because like I already arranged so much. So
yeah, thank you so much, ma'am. Thank you. Sure,
yeah,
we can collaborate in the future.
sure,
Also.
sure,
Yeah.
sure.
Thank
Yes.
you. Thank you so much.
Yeah, thank you so much for having me. Thank you, everyone. Thank you, Ms. Ashika. Thank you, Dr. Suchita. Okay, I'll leave now. Okay.
Yeah,
Thank
thank you. I'll be ending the meeting.